I'd like to start out first by giving a brief introduction on how I like to conduct this review sections, and also to give you a brief scenario about my background, my educational interests, and how can I serve you. With that said, I received actually my PhD in biomedical sciences, and I've been teaching a wide variety of biomedical sciences, biochemistry, pharmacology, physiology for numerous years at professional pharmacy schools, graduate schools, and I'm certified as high complexity clinical laboratory director, and I'd like to share with you my experience. I have 3 of my young men and ladies. 2 of them are physicians, and the third one working her way through medical school, second year medical school. And I like to dedicate these lectures to my former students, to you, and to my young sons, young men and ladies who are struggling to pass on their message and their responsibilities in practicing medicine or getting involved in research in biomedical sciences or teach medical sciences. The way I like to orient this lecture series, Dr. Minarsik and I developed an interest a few years ago to collaborate in providing a highly effective distance education to medical and professional students, and to see how we can assist them. Him and I will start the global medical education series, but unfortunately I did not continue on, and he continued on with his marvelous, outstanding pathology series that's all available online. I gave, I believe, 3 lectures, and due to my commitments, I interrupted the sequence. I still have some obligations and responsibilities to carry on, but I do like to assist all of you by giving a series of lectures in pharmacology, endocrinology, biochemistry, and hopefully I have a complete set of those 3 courses. So what I intend to do for you here is to give you actually a brief introduction on the first chapter that we like to cover. I have a tendency to use materials from more than one source, and as a matter of fact, I developed my own sources when I established molecular endocrinology series and have it completed And I shared it with several publishers and was very well received and accepted by Johns and Bartlett for publication. So I'm going to share this decent preparation that I did. I did very much the same with biochemistry. With pharmacology, I'm working my way through, and I like actually by the end of this course to be able to develop all the figures that I can utilize it freely, with some that I can ask for permission to be used. I'm here to assist you. I'm here to help you out. On the first slide here, you can see actually my website, so please avail the opportunity and visit this website. www.zmedicaleducation.com, and you have actually a couple of lectures in pharmacology, and I believe I asked the IT personnel who assisted me in developing this website to upload also biochemistry lecture, and eventually I will also add endocrinology as well. When we're going to meet, you will receive an announcement from me over the next couple of weeks, and then after that I should be able to commit myself to a regular schedule. Also, you will receive an announcement on the lecture, the topic that I'm going to address in pharmacology, in endocrinology, and in biochemistry and physiology aspects. For me, I love biomedical sciences. For me, I love to see the big picture. For me, I like to pass on the image, philosophy, and dedication to all of you to understand the big picture. When we cover physiology, we essentially covering physiological biochemistry. When we cover physiology, we essentially covering pathophysiological aspects and pharmacological aspects. When we cover basic sciences, we try to understand clinical sciences as well. Be able to appreciate in great detail the overlap, the integration of basic sciences to clinical applications. So please help me to help you to achieve your goal. Last scenario, a fellow of National Academy of Clinical Biochemistry, diplomat of American Association of Clinical Scientists, and I've been elected as a chairman of Education Committee at American Association of Clinical Sciences, and I developed a decent workshop on vitamin D 3 years ago. with my colleagues. With this lecture, I'd like to cover first terminology. I'd like to cover first general pharmacological principles that you should be aware of it before we start to address classes. Pharmacology is highly interesting. When I said before we start addressing classes, I'd like you to understand classes. Names of medications changes periodically, but the classes stay as is. The mode of action of each class stays as is. Prediction of side effects, drug-drug interactions are crucially significant, and it's important to emphasize it. So this will be our focus of addressing pharmacology. You will see that when we address pharmacology, we're covering also basic principles of physiology and biochemistry and pathology as well. I'd like to start out by sharing with you my role model. This is a picture of Albert Einstein and was taken in Santa Barbara in 1933. And he stated, life is like riding a bicycle. To keep your balance, you must keep moving. I'm promising late Dr. Einstein that I will keep moving until the last day of my life. I will pass on my message to you. I'm committed to do that. As I stated to you before, when we address biomedical sciences, I don't see any variation. Between basic and clinical approaches. And I like to visualize it to you as a chess game approach. I like you to see the big picture. I like to be able to stand out and appreciate the movement of every piece. Histology, biochemistry, physiology, pathology, pharmacology, microbiology are the pieces to understand basic sciences and to be able to integrate it into clinical sciences. Please help me to help you to see the big picture and enjoy it. It's highly interesting. When I shared this information with my two sons who are physicians now and my daughter who's struggling to complete basic sciences, they said, Dad, it's with regret actually, after we complete the clinical rotations and the clinical sciences and we're ready to start our residency, we forgot about the basic principles. And we tied on what we're doing. And we felt actually that we were more knowledgeable when we were at a stage of basic sciences. So now appreciate your standing and appreciate your time. We like to be able to address terminology first, to get it out of the way, state what is meant by pharmacology, pharmacodynamics, pharmacokinetics, pharmaceutics, pharmacotherapeutics, pharmacognosy. Be able to go back to the basic principles of biochemistry and address the significance of understanding what is meant by the dissociation constant of a weak acid and weak base, based on the fact that all medications, or most of the medications, are weak acids and weak bases. So it's very important to appreciate the level of their dissociation. It's very important to appreciate how you're going to enhance their excretion versus how you're going to enhance their reabsorption. Understanding of this basic principle will allow you actually to appreciate drugs that can pass through the blood-brain barrier versus ones that cannot pass, and why, and how can we modify it and make it applicable. So we're going to see Henderson-Hasselbalch equation. What does it mean to us in terms of having undissociated species versus a dissociated species? We're going to see the relationship between the pKa value, dissociation constant of a weak acid and weak bases, based on the fact that drugs are weak acids and weak bases. How does that relate to the pH? And which form should be the predominant form? That's not only significant to appreciate the rate of the reabsorption or the rate of the excretion of a drug, but also to appreciate the pathophysiological aspects that can affect this decent relationship. We like to clearly define the difference between pharmacokinetics and pharmacodynamics. How a drug can be handled by human cell. What is needed, what is happening to the drug, and how human body can accept a certain drug via specific receptors that interact with a high specificity toward drugs, toward neurotransmitters, toward hormones. Identify the processes comprising ADME, classify them according to the pharmacokinetics and pharmacodynamics parameters. When we address pharmacokinetics, we're interested in biotransformation. We're interested in bioavailability of the drug. So we have to address phase 1 and phase 2 of the drug. We address various routes for drug administration and what does it mean in terms of their bioavailability. So we should be able to identify the primary location of drug biotransformation and to define what is meant by first pass effect, phase 1 and phase 2. Drugs that are taken orally versus drugs that are taken intravenously. So we're talking about absorption of drugs. Of course, the role of cytochrome P450 isozymes cannot be neglected by all means. So here we're going to address P450 isozymes with respect to drug modulation, drug biotransformation, drug translation, drug-drug interaction, aging problems with isoenzymes, specificity aspects, this outstanding complex system that I actually contribute to some of the work that's been done before. P450, you can see those enzymes moving in a gel structure. Define what's meant by enzyme induction. We're talking about activation of an enzyme and what that has to do with P450 versus increasing the rate of its synthesis. And explain how these processes may affect plasma level of drugs. Drugs that can act as a specific inhibitors for those enzymes. If you induce more of those enzymes, or you reduce those enzymes, drug-food interactions you are dealing with, drug-enzyme interactions. And I can give you right now an example, monoamine oxidase enzyme, and the use of antidepressant agents like amphetamine. It's not recommended for patients to eat cheese because you're increasing the level of the substrate, you're inducing the enzyme activity, and you're demolishing the effect of the antidepressant agent if you do that. So psychiatrists advise their patients not to eat any kind of cheese when they are under antidepressant agents, increasing the enhancing degradation of the biogenic amines. So you're defeating the purpose, actually. Explain how biotransformation can lead to toxic and active metabolites. Distinguish between phase 1 and phase 2 biotransformation reactions. How you bypass phase 1 intravenously, if a drug given intravenously to the liver, and identify how a drug is manipulated as it changes in phase 2. Certain drugs are administered as a prodrug, and in order to meet their efficacy, they have to undergo chemical change to an active drug. So we'll cover that, and I can give you an example right now with vitamin D that undergo hydroxylation reactions in the liver and in the kidney. So if you are giving a patient cholecalciferol, you expect this to be modified in the liver by 25-hydroxylase enzyme and further modified in the kidney by 1-hydroxylase enzyme to give you the most active form 125 called calciferol. Explain the differing effects of metabolism on drugs and prodrugs in cases where metabolizing enzymes are reduced or inhibited. That's very significant because you do not want to defeat the purpose. Then we're going to move on to pharmacodynamics. Pharmacodynamics, we will address how the body accepts a drug via specific receptors, what's meant by agonists that activates those specific receptors versus partial agonists, antagonists that antagonize the production of an effector produced by those receptors binding to a specific hormones or neurotransmitters. Dr. Minarsik, in his pathology lecture, he expressed in a superficial manner the term homeostasis. We like to appreciate this term here when we cover pharmacological aspects, physiological aspects, and biochemical aspects. Maintaining all metabolites constant. How are you going to do that? By a tight control, by feedback regulation, by making sure you have an adequate balance between cholinergic agents versus adrenergic agents. So we're talking about tight control on hormones, tight controls on neurotransmitters. And that is the essence of the following two chapters: cholinergic versus adrenergic drugs. So we're going to deal a lot with that. Specificity requirement. Specific receptors interact with specific drugs. And what does it mean to us? Very, very important protocol to design a therapeutic approaches. You have too much of a drug, you activated this, so you expect huge effect on that. So you may come to the negative side of it. So what can you do? You have to counterbalance it with anti-agonist. You have to relieve the tension of this effect. That's what we like to share with you. Affinity versus efficacy. When you have a drug interact with a receptor, you should be able to define its efficacy and define its affinity towards this receptor. They vary a lot in terms of their specificity. So what are you looking for here? A drug that can work for you. A drug that cannot give you a huge side effects. A drug that can serve the purpose. Get the job done with a minimal toxic effects. When we address those terms, when we address agonists, antagonists, partial agonists, we are addressing essentially a complex protein. That proteins involves receptors, allosteric proteins that you covered in the past when you covered biochemistry and biochemical aspects. So we're going to talk about reversible and irreversible inhibition, looking at the graphs, and we should be able to digest all of these materials. General principles, concepts is very significant at USMLE exam. You expect to see about 25% of the questions devoted to general principles, if not more, about 25 to 30%. The section deals with your understanding. There is very little memorization involved. I urge you to go back and forth and look at the tables presented in this chapter so you can have more and more appreciation about this topic. Let's go through the principles, and next time I will analyze to you in some details this lecture. This lecture, you're going to find it also under my website, www.zmedicaleducation.com. So it's a good starting material. So a drug, it's a foreign body. It's a foreign body that can give you toxicity. Always keep that in mind. I wish the day will come when you limit the use of drugs, because you are taking a risk when you take a drug. Any chemical that affects the processes of a living organism, you are definitely disturbing somehow this outstanding homeostatic status. We are interested here in study of the science of drugs. We're going to touch a little bit on the toxicological aspects. We are interested to see how human body handle a foreign body, a foreign chemical, how it's going to absorb it, how it's going to transform it, how it's going to excrete it. So that deals with pharmacokinetics. What is the exact dose that need to be prescribed to a patient? Based on what? Based on half-life of a drug. So we need to define what is meant by half-life of a drug, elimination rate, absorption rate, see how that follow a first-order reaction, and how we can scale a dose to a drug based on what parameters. How we can enhance excretion of a toxic drug, elimination. You're dealing with electrolytes in the kidney. So we have to understand the terminology of the kidney. We have to provide the proper environment for excretion of drugs. Pharmacodynamics, what the drug will do to the system, how a drug can interact with the system. Receptor specificities, very, very crucial for cholinergic, adrenergic, cardiology system, kidney system. Pharmaceutics, pharmacy schools deal with that. Various forms, formulation of drugs, pharmacotherapeutics, as the name implies. You want to achieve the therapeutic dose aspects of a specific drug. Pharmacognosy is essentially looking at herbal, plant, marine pharmacology as sources of drugs. So you can see that very nicely. Pharmacodynamics what the drug does to the body, mechanism of drug actions, pharmaceutics, drug forms, influence pharmacokinetics and pharmacodynamic parameters, therapeutics, clinical indications for drugs to prevent and treat diseases. You want to be adequate dose. Natural resources, plant and animal drug resources, pharmacodynamics, what the drug does to the body. That's very important. So that collects receptors specificity, collects ligands, small molecules that can bind to a macromolecule. The macromolecule here is a receptor that can be made up of more than one subunit and its specific interaction in order to give you a biological response. With pharmacokinetics, we like to deal with absorption, distribution, metabolism, and excretion. We like to understand each term, appreciate each term, and look at the big picture. And we're going to sum all of this up at the end. When you look at pharmacokinetics, you're talking about stages. How a drug can be absorbed. Is it topical? Is it oral absorption? Intravenously administered? Using patches? And then you're looking at to what extent this drug can be distributed in tissues. Distribution. Is it going to bypass phase 1, go on to phase 2, or it has to go through phase 1 and phase 2? Then you want to get rid of the drug, so it has to be converted into inactive form. You're metabolizing this drug or you're activating this drug. Then you want to get rid of it, so you have to change its ionic state and make it available to be excreted in polar form. If you're interested to help to reabsorb it, so you have to make sure that you have the adequate form to be reabsorbed, and that is the unpolar species. Then the drug can be eliminated either in the urine, bile, or feces. You have a clear definition about the objective of absorption, biotransformation, and elimination in this statement. Measure objective of a drug biotransformation reaction in the body is to make a non-polar drug polar, convert the polarity of the compound from non-polar to make it polar. If it's polar, so yes, it can interact with the electrolytes in the kidney, and we're going to explain that in great detail later, and can be excreted. If it's non-polar, so you're enhancing its reabsorption. When we talk about absorption, distribution, elimination, we talk about pharmacokinetics. When we talk about interaction with specific receptor molecules within the body, we're talking about pharmacodynamics. You have to keep in mind that drugs have to pass specific barriers, cell membranes. So you have to look at the requirement for a drug to pass this cell membrane structure. Certain drugs can pass blood-brain barriers versus others cannot. So what can you do to make a drug more permeable via blood-brain barrier. Common principle that you're going to deal with it when we address autonomic nervous system and drugs that act in this as a class in this class. Avoid alternate sites of attachment and storage. You have to keep in mind that serum albumin is a key carrier. Avoid metabolic destruction prior to reaching site of action. Yes, certain drugs are subject to be destroyed chemically or enzymatically. So they have to be chemically stable at various pH levels. All four of these barriers will affect drug pharmacokinetics. The basic principle of pharmacodynamics is expressed in a very simple manner here. A drug interacts with a receptor to give you drug receptor complex, and that's what gives you biological response. Please, please, please keep in mind you are interfering with the effect of an effector on a drug. You are interfering with a specific hormone receptor interactions. You are interfering with neurotransmitter receptor interactions. And therefore, you tend to modulate the homeostasis process. And then you get the biological response. There are two primary ways in which drugs are eliminated from the body. It can be excreted in the urine or feces. They can be metabolized or biotransformed and then excreted. In some cases, drug may be eliminated in other ways, like respiration, sweating, for instance. But that constitutes a small or minority class. You have to biotransform a drug prior to its excretion. That's a very valid statement, and you have to keep it in mind. We already stated that. You make the polar drugs to be excreted, essentially. And I stated very clearly here, with the process of repetition, a non-polar drug has to be converted to a polar drug to be excreted. We'll go on and talk about biotransformation that results only in making a drug more soluble for excretion. Also produces metabolite that's no longer capable of producing a therapeutic effect. I already give you an example how you convert an inactive or less active drug into an active one. And you see here outstanding presentation for the processes a drug undergoes through it. An active drug has to go through biotransformation, gives you metabolite, and then you change this to more hydrophilic in order to excrete it out. If you want to have it reabsorbed, so it has to be in unpolar species. We talked before about a prodrug that is given and then subject to biochemical reactions can be converted into an effective drug. And this prodrug can be activated in the liver, like vitamin D, blood, inside cells, in various ways. You can cleave a group, you can add a group, addition of hydroxyl or phosphate group. Some examples for you to appreciate that. Chloromorphenyl succinate, so you see an ester bond here, This is giving actual and administered intravenously, so you have bioavailability 100%. Esterase enzyme will cleave off this succinate to give you the most active form, and that's what you see in here. That's the ester bond. Esterase enzyme in the bloodstream cleaves this bond here, give you the most active form, chloromophenol. So you converted the inactive into the less active form. So DAKE drug, an allergic drug, can cause GI irritation when administered. So what can you do? You have to convert it, you have to give it in inactive form first, bypass this. Okay. And then if you do that, so reduced by enzymes in the liver, so you're going to bypass that, and then you have it available for absorption. That's what you see here. So in the middle here is the inactive form. Sorry about that. And then you can convert it to the active form by specific enzyme. Routes, I think I'm going to stop at this stage now because I do have another commitment for online presentation with another webinar at 4 o'clock. So I like to stop at this stage, and I want to thank you all. I see here about 35 attendees. So please encourage your colleagues to join, because it's your enthusiasm that will give me the enthusiasm to continue on and to give you everything I have. I appreciate your time, and this will be processed again and we'll have it available for you guys. But at the meantime, avail yourself for my website, www.zmedicaleducation.com, and you're going to see this presentation there. I may highlight additional issues when I go on and explain that. So please look forward for my next announcement. Thank you and have a great day. Bye-bye.